In this video, we'll look at what happens to carbohydrates and proteins when they enter the small intestine. So this could be either, say this is a long string of carbohydrate here. So that, you can call it a carbohydrate, you could call it a polysaccharide. There are no native enzymes made in the small intestine to cut up polysaccharides. However, this enzyme here, made by the pancreas, is delivered into the small intestine with that job. This is pancreatic amylase. Now, pancreatic amylase cuts polysaccharides into disaccharides, a pair of monomers still attached to one another, a disaccharide off of the chain. Now you've got some disaccharides floating around in the lumen. This is out in the lumen of the small intestine. These disaccharides will diffuse towards the brush border. When they get there, this enzyme here called disaccharidase, so that's a class of enzymes, and this disaccharidase must have as its specialty this disaccharide. Anyway, it will cut that disaccharide into monosaccharides. Now these monosaccharides are imported into the cell along with sodium. So sodium is the driving ion, the import of monosaccharides. And each one of these cotransporters will have its own class of cotransporter responsible for its import. These also have their own class of channels responsible for their export. The import of sugar, just like the import of any other solute, will lower the osmotic potential of the cell. I've drawn in an aquaporin here, even though it's not really relevant to this topic, just to remind you that the import of solute, and in particular sugars, is very important to the water relations between the lumen, the cytoplasm, and then the interstitial fluid behind. You know, because a cotransporter is used, you know because a channel is used, that the highest concentration of these monosaccharides in this process must be in the cell. If it wasn't higher in the cell than in the lumen, you wouldn't use cotransporters, and you couldn't use channels to export it if it wasn't higher in the cell, this concentration, than it is in the interstitial fluid. Proteins enter the small intestine. They've already been cut, I suppose, by the stomach enzyme pepsin, so that they can't have refolded into something toxic when they got here. But here's an enzyme coming from the pancreas to chop up these strings of amino acid, and that is called a pancreatic protease. Now, there are several different versions of pancreatic protease, and they all have a different subset of targets within this protein that they're going to cut. So you're going to get smaller chains called oligopeptides chopped by these enzymes. And the oligopeptides are still too large to be imported into the brush border cell. So another enzymatic process is required, and that is the job of these oligopeptidases to turn oligopeptides into amino acids. And the amino acids have to be co-transported into the cell via sodium-driven. Notice that this means there must be more amino acid concentration inside this cell than out. Some amino acids are charged, some amino acids are neutral, but just in general, you can assume there's more amino acids in the cell than there is out in the digestive lumen. This is the lumen, this is the cytoplasm, this is the interstitial fluid, this is the apical surface, this is the basolateral surface. Okay, so these amino acids are then imported, and then they can be exported through these channels into the interstitial fluid on the basolateral surface, and then The bloodstream is down here. Here's a capillary, and this stuff will enter via mechanisms we've talked about earlier in the class when talking about the circulatory system.